Welcome to Study Live. We are seeing the chapter Biomolecules, and in this session we are going to start about classification of lipids. Now these lipids, they are classified into three types. They are divided into simple lipids, compound lipids and derived lipids. So first we are going to see about is simple lipids. Simple lipids. Now, first is the simple lipids. These simple lipids, they are esters of fatty acids and various alcohols. They are esters of fatty acids and various alcohols. So these simple lipids, they are basically what all is included in this. In this we have fats and waxes are included. Fats and waxes are included in simple lipids, are types of simple lipids. So first let us see about the fats. First we are going to see about fats. Now fats, they are esters of, they are triglycerides. They are esters, which means here they are triglycerides. They have got three fatty acids attached to, they have three fatty acids attached to glycerol, Now in this we are going to see there are three fatty acids attached to glycerol. They are saturated fats or unsaturated fats. The saturated fats are the animal fats which are solid at room temperature, and the unsaturated fats over here are the plant fats which are liquid at room temperature. They are plant fats which are liquid at room temperature. So they are nothing but they are triglycerides, where we have three fatty acids attached to a molecule of glycerol. So when we compare this with the waxes, so what is the difference between over here between fats and waxes? They are esters of fatty acids with long chain alcohols, So here the difference of the alcohol over here is, in this we had glycerol and in this we have long chain alcohols. Let us see the difference between fats and waxes. These fats, they are the nutritional reserve food material. Reserve food. In animals, it is stored in adipose tissue. In animals, it is stored in adipose tissue, and in plants, example, we can see in case of seeds, it is stored as a reserve food material which is used for the germination of embryo. It is used in the embryo stored for, it is used for seed germination. Now in animals, it is also present around internal organs in animals, and it is protecting from mechanical shock against injury, mechanical shocks and injury. So it is protecting from mechanical shocks and injury. So this is the about the fats, where it is the nutritional reserve food in animals and plants. And in animals, we can see it is stored in the adipose tissue as fat droplets, whereas around the internal organs, it is protecting from mechanical shocks and also preventing injury. Let us see about the waxes. These waxes, they are insoluble. They are not soluble. They are also solid at room temperature, such as you can see bee wax, which is present in bee hive. Now these waxes, they are also present in plants as well as in animals. In animals, these waxes are present in fur and feathers. They are present as a coating on fur and feathers. Fur and feathers. And these waxes, they are insoluble in water, so it is helping protecting their fur and feathers, whereas in animals we have seen it is present, like you can see hair of the dogs, which is the fur of the dog, and in birds it is present in feathers. In plants it is present on the epidermis, which is the outer layer of leaves. Epidermis of the leaves has a wax coating which is made up of cuticle. Then it is present in the outer layer of stem. It is present as a coating on fruits. So these waxes are present there. So this completes our first simple lipids. Our second is about the compound lipids. Compound lipids. Now, as the name suggests, compound lipid, it is containing other group in addition to fatty acid and glycerol. What is the structure of compound lipid? So here, two of hydroxyl groups of glycerol, are esterified. They are esterified with fatty acid. While the third hydroxyl group is attached to either phosphate group or to a sugar molecule. Now, when it is attached to a phosphate group, it is called as phospholipid. When it is attached to a sugar molecule, it is called as glycolipid. So here we have an additional group which can be a phosphate group or it can be a sugar group. So compound lipids have got phospholipid, glycolipid. Now in example, lecithin, which is a phospholipid. This lecithin has nitrogen atom in addition to phosphate group. Now this phospholipid which is present, in the cell membrane. If we see this phospholipid, it is present in the cell membrane. Where we can see there is a phospholipid double layer, bilayer membrane, which is called as fluid mosaic model. Here we have the polar hydrophilic head. This is the polar hydrophilic head. Whereas over here we can see, here we have non-polar hydrophobic tail. So these are the non-polar hydrophobic tail. So this is the phospholipid bilayer which is present in the cell membrane. This is the fluid mosaic model explaining this bilayer of lipids. So the next is about the glycolipid. Now glycolipid contains sugar atom. This glycolipid contains sugar in addition to, now this sugar can be, it is different in plants and animals. In plants, the sugar present in glycolipid. The sugar present in glycolipid is galactose. Whereas in animals, the sugar which is present in animals, the glycolipid which is present, this glycolipid is called as cerebroside. It is called as cerebrosides. And the most common cerebroside which is present, the common cerebroside which is present is called as galactocerebroside. Galactocerebroside. So if we draw the structure of this compound lipid, we can see that over here two of the hydroxyl groups of this molecule of glycerol. So if this is the molecule of glycerol, two of the hydroxyl groups, see this hydroxyl group, one is gone, the second one is gone. So two of the hydroxyl groups, they are getting esterified by a fatty acid. So this is the fatty acid which is getting attached over here. Two of the hydroxyl groups, they are getting esterified by fatty acid, while the third hydroxyl group is either attached to a phosphate group or to a sugar molecule. And in case of example which is lecithin, we have got, in case of lecithin, we also have a nitrogen atom which is attached along with the phosphate group. So this is the example for compound lipid. So in this classification, we have seen about simple lipid and compound lipid. So there is one more which is derived lipid. The third classification is going to be derived lipid. Now this completes our topic on classification of lipids. So for more videos, please stay tuned with Study Live. Thank you.